Here at the Field Museum, we have 2 million plant specimens in our collections to help us solve big questions in biology, especially questions about the interrelatedness of species. Let's look at the case of the liverwort genus Frillenia. Sidebar. Liverworts, along with hornworts and mosses, are part of a group of non-vascular early land plants called bryophytes. Bryophytes have to be really small in order to get water, because unlike ferns and trees, they have no internal vascular system to transport that water. So that's what we're talking about, tiny, tiny bryophytes. Okay, back to the research. We had some suspicions about 2 Frillenia species, Rostrata and Falciloba. We wondered if there were some spies in our midst, liverworts that had just been called Rostrata and Falciloba because nobody had ever given them a really close look. Our goal is to resolve the long-held identities of these 2 cryptic groups, basically to figure out if the plants currently in these species complexes actually belong there. So we gathered a whole lot of collections from the museum's herbarium and investigated them by pitting them against each other. We have 2 lines of attack, morphological and genetic. On the morphology end, we look at the plants really closely to understand what they look like on a microscopic level. We made a huge list of each one's morphological characters, recording branching patterns, the margin of the stem underleaf, how many cells were in the leaf lobes, and the length of the median cells. Tiny measurements for tiny plants. We needed to see if any of the liverworts look different from each other under a microscope. But physical appearance isn't the only way botanists can tell plants apart. We can also look at their DNA, the language of cells, to see how related individuals are on a molecular level. We start by taking the cleanest, youngest buds of each plant and snip them off until we have enough plant material for testing. Then we extract DNA from the liverworts. I like to use a power drill to grind Frillenia into powder. It disrupts the cells and increases surface area, and it's also really fun. We add solvents and enzymes that will remove proteins, lipids, and RNA from the mixture until all we're left with is pure liverwort DNA. Next, we use a process called polymerase chain reaction, or PCR. With PCR, we can amplify our purified DNA to create copies of a specific fragment. PCR has 3 stages. First, we separate the 2 strands of the DNA double helix by heating them up so they denature. Second, we bring the temperature down so our primers can anneal to the strands. We're using primers that are known to be useful for this kind of liverwort research. In the 3rd stage, we raise the temperature so that DNA polymerase can find our primers and copy the DNA. We repeat these 3 steps for exactly 2 hours and 36 minutes, and in the end, we have millions of copies of DNA. Then, we take the DNA we've replicated in PCR and run it through a sequencer so we have real chains of nucleic acid sequences to play with. If we really do have spies in our midst, we should be able to figure it out by looking at the arrangement of nucleotides in their DNA. Even if the spies are morphologically identical to the average Frillenian citizen, their genetic information will be different from the liverworts that actually are Frillenia rostrata or Falciloba. When we're finished with this project, we'll understand so much more about the evolutionary interrelatedness of liverworts, and also the best ways to identify biological double agents, from morphology to genetics. We still have a lot more work to do on this project, and if you want to help us answer these and other big questions, you can go to microplants.fieldmuseum.org to help botanists at the Field Museum solve biological mysteries. Thanks!